So looks like everybody's just about settled down. So if everyone can just grab a seat, and we will get started with our next session. As I know, I saw a number of you catch Marley and Uva and Gwen at the break. Again, if you guys want to grab them at any time and ask any questions, I know that they'd be more than willing to answer those. And our next session is going to be on, hold on, I have the wrong date. I have my thing upside down. Where are we at? 20th? 21st. Ah, there we go. So, sorry, I had my paper upside down. The next section is going to be on therapy in focus, and our first speaker is Isabelle from Paris, and she's going to give us an update on the USHStat, Myo7, and other clinical trials. We have a bunch of stuff that's close to clinical trials or just about in clinical trials, so this will be good stuff.
Thank you, Mark. And okay, excellent. So thank you very much for this very exciting meeting and the opportunity to share with you some of our data. Actually, I'm just going to give you a few examples, and there's a lot of very exciting talks coming up with all other therapeutic developments. So if you want to have therapeutic approaches, and I'm going to talk essentially about retinal disease. I'm a retina specialist, so I'm going to talk essentially about the retinitis pigmentosa part of Usher syndrome. Is either you want to try to stop the progression of the disease or to restore vision. To stop the progression of the disease, there were a very exciting development in the past years, aiming at treating the genetic cause. So that could be gene therapy, either by replacing the gene, the defective gene, by a new normal genes, or correcting the genes. And you'll hear in other talks about oligo antisense nucleotide, about CRISPR-Cas, about read-through drugs in order to correct the disease. So regarding Usher, the problem is if you want to bring the normal genes to replace the defective genes, you need to have a vector that will deliver the genes to the targeted cells. So you usually use viral vectors, and the first viral vector that were used are adeno-associated viral vectors called AAV. Unfortunately, most of the genes that are mutated in Usher are too large to be packaged in AAV, or they need to have some special tricks in order to be delivered by AAV. So there's one trial in which our center in Paris is involved, which is sponsored by now the pharmaceutical company Sanofi. And this trial has been using not AAV but lentiviral vectors that are able to package larger genes, up to 10 kb, which is quite large. Some Usher genes are even larger than that. So the idea is this virus, this lentivirus vector, it's called IAV. It's the virus that cause anemia in horses, and it's not a pathogen in humans. We are not horses. And there was preclinical studies that documented the safety of the delivery of this viral vector. And currently there's two trials that aim at packaging and delivering through this lentivirus a normal gene. One is for ABCA4, and it's another disease which is called Stargardt disease. ABCA4 is the long gene. And the other one is Myo7A. As you know, Myo7A mutations cause the most common form of Usher type 1. So prior to that, there were experiments in animal models, and this is a slide as an example of what was performed in monkeys. If you want to use a viral vector, you have to prove that you're indeed able to infect the targeted cells. So what Sanofi did is on non-human primates, they delivered the viral, the EA1V viral vector subretinally, and then the virus was not expressing a gene to treat, but a gene that was able to made a protein that could then stain the cells. And by doing that, they were able to show that their AA1V was indeed able to infect cones and rod photoreceptor and also RPE. What they also did is they performed subretinal injection in non-human primates in one eye. They injected one eye and subsequently the other eye, or they re-injected the same eye, and they could document that it would not lead to major inflammation. So that's when the trial started. So the trial is called USHStat, and it's really aiming at one single injection underneath the retina of this viral vector delivering the normal Myo7 gene. This is a trial that is taking place in 2 centers, Portland, Oregon with the Casey Eye Institute in the US, and our center in Paris. So the design of the trial is a dose escalation. So it started by patients that had severe disease with the lowest dose, because the first point is to document that it's safe, that it doesn't lead to major side effect, because that's not what you want, although it was safe in the preclinical test. So the first patient had very severe retinal degeneration linked to Myo7 mutations, and then the dose was increased while we were documenting safety. So far, 9 patients have been involved in both centers, and with a nice safety profile, which is provided by Sanofi. I forgot to say I have no conflict of interest. I'm not paid by Sanofi. And 9 patients involved. There were several side effects, and actually one side effect in the latest patient was panuveitis, which means after the surgery there was inflammation in the vitreous. So after this inflammation, everything was resolved, and most of the patients recovered at least the vision they had prior to the surgery, to the subretinal injection. But when the independent safety committee discovered that there was one case of the 9 patients who developed severe inflammation, they wanted to pause the trial. So up to now the trial is on pause. Sanofi did further tests, and the safety profile was re-studied with half of the highest dose, and the trial should restart before the end of 2018. So the good thing is, we have limited data so far on this patient. As I was telling you, this is mainly looking at safety, but there's some efficacy that are evaluated based on visual acuity and visual field. And on these slides, I put some results of the visual field for the 9 patients. All the patients that were treated recovered their baseline visual acuity, and even some of them have after the surgery and after in this graph, for some patients were followed for 150 days. Some patients are starting to have a difference between visual acuity in both eyes, but very little patient. So, yeah, sorry. Oh, excuse me. Yeah, sorry. So I will slow down. So this slide was to show you that the majority of the patients recovered their visual acuity, the visual acuity that they had prior to the injection. And then these patients are followed, and what we would like to document, because only one eye has been injected, is to see with the follow-up whether the injected eye behaved better based on visual acuity and visual field. And in some patients, we are starting to see some differences, but it should be taken with caution. So again, the trial has been on pause for a year now to have more safety data on the preclinical side, and it should resume before the end of this year. So that was for Myo7 gene replacement, and you'll hear about other gene therapy approach later on. So I wanted to now touch upon a few other trials that are either at the preclinical stage or will be starting very soon or already started. One is independently of the gene. You want to stop the disease. You know that for the retina, the rod photoreceptors are degenerating first, then the cones. The cones are more precious photoreceptors because they are responsible for our daylight vision, our reading, our color vision. And one other approach would be independently of the genetic cause, can we save the cone? Can we stop the cones to degenerate? And this is some work that was done by my boss, Professor Sahel, for a long, long time. His idea was that not always in Usher, but in other type of retinal degeneration, the mutation is really carried and expressed only by rod photoreceptors, but the cone degenerates secondarily. And his idea was that not only rods are important for night vision, but they are also able to synthesize, to make factors, proteins, that are really critical for cone survival. And when rods degenerate, then cone degenerate. So there was a huge research going on in our institute to identify this rod-derived cone viability factor. And one was published in 2004. It was shown in different animal models that it was indeed able to stop in few animal models with rod cone dystrophy, with retinitis pigmentosa. It was documented that their cones could be, the degeneration of their cone could be slowed down or stopped. And even that the structure of the cones, the morphology of the cone was even improved. And there would be a clinical trial, initially in a very selective genetic group of patients, starting in 2020, the group of patients that mirror the preclinical studies on the animal models. But we hope that with the proof of concept that we're indeed able to deliver, by delivering this trophic factor or the CVF, we can stop the cone to degenerate, then it should be widespreadly tested, including in patients with Usher syndrome and retinitis pigmentosa. So that was the axis of stopping the disorder, either by gene editing, gene correction, gene therapy with the current Myo7 clinical trial, or protecting the cones. But now you know that there's other type of therapeutic approach to restore the vision when you no longer have photoreceptor and especially the cones to degenerate. So Professor Zrener will speak about artificial retina, so I will just really briefly speak about that. And the idea of artificial retina is when you no longer have photoreceptors, only photoreceptors are able to be activated by light to generate an electrical signal at the optic nerve towards the brain. And so the idea of the artificial retina, and another approach I will mention later on, is to restore the fact that no longer cells are photoactivable and able to send information to the other retinal cells. Other retinal cells, such as the bipolar cells that are connected directly to the photoreceptor, or the ganglion cells that are the third neuron forming the optic nerve and sending the vision to the brain. And these two types of cells usually are not degenerating during artificial retina. So there's different type of artificial retina. Underneath the choroid, underneath the retina, but underneath the layer of support the retinal pigment epithelial, or subretinal, or on the top of the retina. In our group, especially the group of Serge Picot in our institute, has been working with Stanford and Professor Palanker to develop special artificial retina with little diodes here that are completely independent from each other. And the idea of these diodes is to be subretinally placed surgically, and then with the camera looking at the environment, coding the information and sending back the information to these microdiodes by infrared light to encode the vision. And this is something that, so you see the implant underneath the retina. This is something that has been tested in primates. And so this is a bit innovative in a way that there's no connection for this little chip with diodes, because it's just activated by this infrared light. And there's currently a trial, which is not yet for retinitis pigmentosa, but we hope it will come, in geographic atrophy in 2 centers, one in Paris, one in Pittsburgh, Pennsylvania, on geographic atrophy. And the idea is, so geographic atrophy in age-related macular degeneration. And the idea with this little chip that can reach, we hope, 1,000 electrodes, it's to prove that we are able to restore fine vision in these patients that have only macular involvement. Because if we can do that on patients who have only macular involvement, then you can also restore that when not only the macula is involved, but also the peripheral retina. The last trial I want to mention is optogenetics. So what is optogenetics? So again, when you have retinitis pigmentosa, you lose the cells that are able to be, the only cells that are able to be activated directly by lights into the retina. So the idea of optogenetics is not to restore photoreceptors, but to transform the other cells, bipolar cells or ganglion cells that are not affected by the disease, and to make them photosensitive. And the idea is you have some protein in algae, ancient protein in algae in the sea, that are able to be activated by light and directly open a channel and initiate a signal linked to the light. So the idea, so there's 2 types. One is halorhodopsin and one in channel rhodopsin. So the idea with optogenetics, it's a sort of gene therapy. It will be injection within the eye. It doesn't need to be underneath the retina, because you want to target either the retinal ganglion cells or the bipolar cells, to inject the genes of this channel rhodopsin, this protein that are able to be activated directly by light and generate the signal. Unfortunately, this channel rhodopsin can be activated by natural light, so this type of approach will need to be coupled with specific goggles that will transform our environment and generate light for this, the approach that is developed in Paris. It will be red lights in order to activate this photopigment that will be mostly in the ganglion cells. So again, the group of Serge Picot has performed testing on mice, showing that they were able to indeed infect ganglion cells and generate an electrical signal by stimulating this channel. It was also performed in macaque, in monkeys, and this is the intravitreal. It was proven that just injecting inside the cavity of the eye, not underneath the retina, but inside the eye in the vitreous, the channel rhodopsin was distributed quite widely, so it could give a larger vision. So the trial is supposed to start anytime soon. It has been accepted in France. There will be other other centers in the UK and in the US, and we just got the approval. And so the idea would be one single injection in the eye and testing this device with the goggles to see if there's any possibility to photoactivate and restore sun vision at the cellular level. It's a sort of similar approach than the retinal implant, but you really go directly to the cells trying to have maybe hopefully a better refined visual restoration. So there's also a very exciting development also in the group with one of the mathematicians that works in Paris and in Pittsburgh, is to try to combine optogenetic or implants with a precise coding of the visual signal by a specific camera, in taking into account how our brain is able to analyze the space with the contrast and the speed, and to try not only to improve the device that is delivered to the retina, but also improve the way the information is treated by a camera and sent to this different approach. And so this is to be continued, and I want to thank you for your attention. I'm sorry, I spoke too fast.
So thank you, Isabelle. As I mentioned before, Isabelle is available to answer any questions that you might have.